DNA replication. We're just copying one gene, the gene that codes for the needed protein. Second thing we need, this is what's going to be copied. We're going to make our new copy with mRNA codons. So they have to be available in the cell, which we call free, which means a healthy cell that has good nutrition should have all the components and be able to make nucleotides to make RNA. They're going to need free RNA nucleotides. Right? To build the mRNA, and actually the rRNA and the tRNA also. And third thing you're going to need is enzymes. This time, there's another complex of enzymes for making mRNA and tRNA and rRNA, but we're just going to call it RNA polymerase enzymes. Polymerase means building polymers of RNA. It's actually a whole complex of different enzymes. Different enzymes to do different jobs. Your textbook gets into a lot of detail and tables, but I'm not going to test you on all different kinds. Just know that you need enzymes to make the RNA. Okay, let's say our mRNA copy has been made. Now we're going to have to get a shopping list together for translation, the second step. In the first step, the cell transcribes DNA language into mRNA language. That's why it's called transcription. The cell copies the DNA into a working copy of DNA, so it has to transcribe DNA into RNA. The second step, you're going to translate RNA into a chain of amino acids. So now you're going to need mRNA, which is going to be red. That's the working copy of the DNA. You're going to need rRNA, that's your ribosome. You may remember I wrote this on the board last time. And you're going to need tRNA. That's the one that does the translating. But it has to bring proper amino acids into place. So something else you're going to need is free amino acids. If the cell is healthy, it should have all the component parts to make amino acids. We're not going to get into it in this class, but you're aware that some amino acids are essential, some are non-essential. The essential ones you cannot make from components, you must ingest them. And the ones that are non-essential, your body can make with components from other molecules. So eat your rice with beans, because rice will provide some of them, beans will provide others. You'll have all your essential amino acids. So you're going to need free amino acids in order to build the chain of amino acids. And guess what else? You're not done. You still need your enzyme complex, and we're still going to call it RNA polymerase. So what happens in transcription? Cell converts DNA into mRNA. What happens in the translation step? The cell translates the mRNA into a chain of amino acids. And what does the chain of amino acids become? A protein. Transcription happens at the nucleoid. Translation happens on a ribosome out in the cytoplasm. You went from DNA to RNA to protein. Okay. This, I don't even remember if I put it on your final exam now, but last semester this was on your final exam. Not your final exam, their final exam. And I was shocked at how many people got it wrong. Even though they understood transcription and translation, they weren't seeing the big picture. And this is the big picture. Your textbook calls it the central dogma. I hate that terminology. Dogma is what everybody thinks it is. Dogma is the current belief. Like many, many hundreds of years ago, we thought the earth was flat. That was the central dogma. Well, now we think the earth is round. Now that's our central dogma about the earth. Well, there's a central dogma about how genes work. And this is it. The gene, which is your DNA, is copied to RNA, and the RNA is translated into protein. So genes code for proteins. But in order to get there, we have to make the working copies. So this was the big picture that students missed last semester. DNA is made into RNA, and RNA is translated into a protein. So this step is transcription, and this step is translation. Just in case that's a final exam question, I don't want you to get it wrong. DNA is not what we make proteins from. We make proteins from RNA. But you can't make RNA unless you have DNA. So you transcribe your DNA into RNA, then you take your RNA and you translate it into protein. And that's how genes code for proteins. Then the proteins assemble into tissues, cells and tissues and tissues and organs and organs and organisms, right? Okay. That's the...
Oh, don't reset.
This part?
Your little,
That part.
you're fine right there.
Okay. And we got to do induction and repression. So I could go through all your PowerPoint slides, but we don't have much time, and I think I'm faster without the PowerPoint.